Hello, my name is Simon Hemelaar, and in this short video I will demonstrate the biocompatibility of fluorescent nanodiamonds. We want to use nanodiamond particles as intracellular sensors. Of course, we want the cells we investigate to be as happy and healthy as they always are. Therefore, it is important to measure the impact of diamond uptake on cells. In this research, we use HeLa cells, since they are also a commonly used model in cancer research and have been shown to readily take up nanodiamonds. To find out how the cells respond, we measured the metabolic activity, which is a measure for the vitality, the production of free radicals, and changes on the proteomic and genetic level in different conditions. These conditions include different concentrations, different time points, different sizes and shapes of nanodiamond particles, and also we included an aggregated status. We measured everything after an uptake period of 5 hours. As a positive control for cell damage, we used hydrogen peroxide. Here we have visualized the uptake of fluorescent nanodiamonds with a diameter of 70 nanometers, both directly after uptake and 24 hours later. The cells are stained with phalloidine fits, a green label for their actin cytoskeleton. The nitrogen vacancy center gives the diamonds their fluorescent signal and is visualized in the middle panels and in the right panels in red. In these panels, we have used larger and rounded nanodiamond particles to incubate into the cells. Normally, nanodiamonds have sharp edges, which is why we also wanted to investigate the influence of the shape of the diamond. With the images shown before, we were able to make an estimate of the amount of uptaken diamond particles, using a software protocol designed by my colleague Felipe Perdona. Obviously, a higher concentration of diamond particles fed to the cells also results in a higher amount of uptake. In the other samples, we see approximately the same amount of uptake. MTT is transformed into a colored product when the cells have an intact metabolism. The absorbance of this dye gives a measure for the viability of the cells. Everything between 80 and 120% of the control situation is considered to be normal viability. We use 2 hours of incubation with different concentrations of hydrogen peroxide as a positive control for cell damage. Using a compound called DCFDA, we can measure the total amount of generated oxidative radicals. Cells generate more radicals when threatened, and this can be used as a measure of stress for the cell. In cells where the stress-inducing hydrogen peroxide is added, a 100-fold higher response shows that the method can measure the produced radicals. The inset in the graph shows what cells which generate the fluorescent metabolite of DCFDA look like under the confocal microscope. Cells themselves have, of course, defensive mechanisms to save themselves from oxidative damage. An O2 radical can be converted to hydrogen peroxide by superoxide dismutase, which is further metabolized to either water and oxygen by catalase or to water by glutathione reductase. When there is too much oxygen radicals, caspase 9 is upregulated in cells, which starts a cascade to upregulate caspase 3. This in turn leads to programmed cell death, otherwise known as apoptosis. Here we look at the genetic changes of the scavenger-related catalase, glutathione reductase, and superoxide dismutase, and the apoptosis-related caspase 3. The relative expression is the transcribed messenger RNA compared to the control situation, measured using real-time PCR. This is a measure for the intentions of the cell as it responds to the current situation. The RNA samples were collected directly after incubation with diamonds, 24 hours after diamond uptake, or directly after incubation with hydrogen peroxide. The most important finding here is that in normal working concentrations of 1 microgram per milliliter for diamond-based sensing, no difference of RNA transcription can be found. The more evolved response of the cells can be measured with the protein level, As mRNA is transcribed, it can be translated by the ribosomes for the production of new proteins, or it can be broken down by the cell through a homeostatic process. The protein expression was tested using Western blot and compared to the control. No significant differences were found after diamond uptake compared to the control. I hope to have convinced you of the compatibility of nanodiamond particles in HeLa cells. This research will prove invaluable when performing intercellular quantum measurements using fluorescent nanodiamonds. In none of our experiments we could pinpoint adverse effects of diamonds to the cells. Nanodiamonds were already shown to have no negative influence on the viability of other in vitro models. In this research we analyzed the cell's response on a highly detailed level, supporting these claims. Thank you very much for watching this video. If you liked it, please check out our other videos at www.bioanalysis.nl/videos. Thank you very much.